The goal of life, biologically speaking, is to achieve genetic immortality. And since the cell, the organism, isn't going to last forever, it has to make another vessel to carry those genes on to the next generation. Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this first of our genetics unit notes cast, we're going to be talking about the cell cycle and how the cell gets ready to do that all-important thing, and that is make another set of genes and another cell to carry those genes on to the next generation. So where do lives begin? They begin with a single cell. Little tiny thing. It's actually smaller than this, but you get the point. And now you're tens of trillions of dynamic cells that together can write poetry and ride bikes and sing songs and do all those great things that you can do. And what we have to take a look at is how we can get from that one single cell to those tens of trillions of cells that make up your fabulous selves. On the simplest level, that original cell is going to have to basically just divide and divide and divide and divide. These first divisions are kind of interesting in that the zygote here, and here's the size of the original zygote, it's now 2 cells. But when those 2 cells divide into the 4 cells, the zygote kind of stays the same size, the entire structure all the way through this morula stage here, all kind of is the same size. That's because the cells don't grow in between each mitotic division. But reproduction amongst simple single-celled organisms like amoebas is one of the functions of mitosis, asexual reproduction. It's also used for growth, obviously. We started out as 1 cell, and now we're trillions of cells, and that's all mitotic. And in that process of growing up, of course, we fall off our bikes and skateboards and leave a little skin behind. And so we have to grow some new tissues for repair, as well as growth and reproduction. Some of the cell parts we'll be talking about as we talk about mitosis are the nucleus, where we'll find chromosomes, which are made of DNA, and the cytoskeleton, which provides the scaffolding for the movement of the parts of the cell, the movement of the cell itself. But what we're concerned with primarily is the movement of those chromosomes. Centrioles are part of the cytoskeleton. They are kind of microtubule organizing centers. But we're going to find spindle fibers in all cells that are going through mitosis. When it comes to the nucleus, its main function is to protect that DNA. Out in the cytoplasm, there are nucleases, enzymes that will chemically break down the nucleic acids that they bump into, and we don't want our DNA to be broken down by those nucleic acids any faster than they already are. Structurally, it is made of a nuclear envelope that is a double membrane, and there are some spots in the double membrane that create these, they're surrounded by proteins, create these pore complexes, they're called, and that's going to allow large molecules like RNA to be able to leave the nucleus. Here's an actual electron micrograph of the nuclear envelope itself, and you can see where the pore is located. Taking a moment to review the cytoskeleton. Functionally, it's to provide structural support for the cell, giving it its shape. It's also going to allow for the organelles to be somewhat anchored in position. They're made up of protein fibers, and they make microfilaments and intermediate filaments and microtubules, although we're mostly concerned with the microtubules. The cytoskeleton is also involved in motility, moving that cell around from one place to another. The cilia and flagella can be thought of as really extensions of the cytoskeleton. And in moving around and in maintaining the shape of the cell and going through phagocytosis or endocytosis and exocytosis, it's going to be involved in the regulation of the cell activities themselves. Another diagram here, just so you can see the parts. The blue microtubules are what we're mostly concerned with. Well, they're blue in the diagram down here. These long rods of tubulin are the microtubules. Up here in the scanning electron micrograph, it's actually the green parts that make up the microtubules. And you can see how they are closely associated with the center of the cell and with the nucleus. But they are shot through the whole cell. Cell division in animal cells involves centrioles acting as microtubule organizational centers. They're going to create the spindle fibers. And the spindle fibers create tracks, or are used as little tracks for the motor proteins to drag chromosomes along. Speaking of moving chromosomes around, we need to make some daughter cells here. So we have to not only just move the chromosomes through mitosis to get a complete set of DNA in each daughter cell, but we also have to get all of the other cellular machinery into each daughter cell. And this process of cytokinesis, moving those materials through the daughter cells, is going to utilize the same parts of the cytoskeleton. Here you can see in this stained cell of a kangaroo rat epithelium, you can see that the cytoskeleton does extend through the entire cell, and so it's going to provide a good network to move those organelles and other cellular structures around. Our next video is going to look at each one of the active stages of mitosis in quite a bit of detail, but I'll run you through them real quickly here so we know kind of where we're heading. We're talking today about interphase, and really when we talk about cells in general, we're talking about a cell that's in interphase. We call this resting phase. It's not really resting phase. The cell is doing quite a bit of stuff, which we're going to talk about here in a second. But when a cell goes through active mitosis, it's going to go through a series of steps to ensure that a complete set of DNA and all of the organelles and other material that are necessary for each daughter cell gets divided into those daughter cells the way that is necessary. So first, the chromosomes are going to form. They're double chromosomes now, and the spindle fibers start to form. They're going to attach to, the chromosomes are going to attach by their centromeres to the spindle fibers and then line up across the equatorial plate, across the middle of the cell. Spindle fibers are stretched all the way across the cell now. And then in anaphase, the chromosomes, the double chromosomes break apart at their centromeres, and each individual chromatid moves towards its pole of the cell. When we get to telophase, this is where the nuclear envelope starts to reform and the chromosomes start to break apart back into chromatin. We're also going to get cytokinesis taking place here, so the rest of the organelles that need to be in the daughter cells get to where they need to be. The whole process here goes interphase, prophase, metaphase, anaphase, so we usually call it IPMAT. Interphase makes up the longest part of a cell's life. This is when the cell is doing what the cell is supposed to be doing, living, learning, loving, jumping up and down, making RNA and synthesizing proteins, basically. If it gets the right signal, it is also going to get ready to go through mitosis. In a typical high school biology classroom, the life cycle of a cell is described like this. A cell is formed by mitosis, it gets bigger, grows, and matures, and then it divides as well. So all your cells are going through all of this division, right? They're going through each one of the stages of interphase, and then each one of the stages of mitosis, and dividing and dividing. That's not untrue. That is happening in epithelial cells and in white blood cells and in stem cells. But the truth is, most of the cells in your body have grown. They come from mitosis. They've grown, they've matured, and that's kind of it. They don't go through those other stages. This is what happens in muscle tissue and in nervous tissue. Some cells some cells that have gone into this resting stage can revert in things like your liver. Some of those cells can go back and start to divide again. Again, interphase is a busy time. There's a lot taking place. It's divided into three distinct phases. The first one is called G1, or the first gap or growth phase. This is when the cell is doing all of its normal metabolic processes, synthesizing all those proteins, doing all that stuff that it does. It's going to be growing during this phase as well. And this is where most cells are going to kind of stop and enter the resting phase, or G0 phase. Now, it's not resting, it's still going through all of its metabolic processes. It's just not getting ready to divide. If it does get the signal to divide, then the cell enters the next phase called the S phase. And this is S for synthesis of DNA. And so this is when replication of the chromosomes is going to take place. After that, the cell goes into the second growth, or the second gap, the G2 phase. During this phase, the cell is getting ready for the rest of division. So we're going to get a little bit more growth as the cell produces the organelles and all the other stuff that needs to be divided into each of the daughter cells. When you look at a cell to see that in interphase, what you're really going to notice is that it has a nucleus, that it's well defined, and that the DNA is in chromatin form. During S phase is where we really start getting into some genetics, because this is when we're going to get a full set of DNA for each one of the daughter cells replicated. In a human cell, that's quite a bit of DNA. You've got about 3 meters of DNA in each one of your 10 trillion cells, so that's quite a bit. And each one of the daughter cells has to get a complete set. There is quite a bit of room for error. And we make about 1 error per 100 million bases, and that works out to be about 30 errors per cycle. And this, it sounds like a lot of mutations, but when we start thinking about the volume of genetic material there is, it's actually really, really accurate. In order for the replication of S phase, or the transcription and translation during the G phases to take place, DNA has to actually be able to unzip. That way we form that replication bubble or the transcription bubble. And if you don't know what I'm talking about with these things, go back and watch those videos. But DNA doesn't exist in just a plain old double helix. I mean, it's just too long, 3 meters, way too long to fit inside that tiny little microscopic nucleus. So the DNA is actually, to shorten it a little bit, make it a little more manageable. The DNA is wrapped around these histones. The histones, they're little proteins that act like spools, so that you don't have all of this loose stringy DNA all over the place all the time. And so you get like this kind of string of histones, looks a little bit like a string of pearls. Those get coiled up to form chromatin, and this is the form of DNA that we find during interphase, because it's pretty easy to unwrap the DNA off of the histone spools and form a replication bubble. But the problem is that it's still, as chromatin, too long and too chaotic to make sure that we get one full set of DNA in each daughter cell. So before mitosis takes place, that chromatin is going to condense down into a mitotic chromosome. That's a smaller package that makes it a lot easier to distribute the genetic material equally. And this is what's taking place in the first stages of mitosis, going from interphase into prophase. We see that condensation of chromatin into chromosomes. Here's a scanning electron image of double-stranded human chromosomes getting ready to go through mitosis. This mitotic chromosome then is a duplicated chromosome. It's the result of replication. So instead of one strand of DNA, we actually have 2 strands of DNA that shorten down into individual, what we call sister chromatids. The 2 sister chromatids have a little narrowing, which we call the centromere. This is where the kinetic cores and the point of attachment to the spindle fibers is going to take place. But it's important for us to remember that since they are the product of replication, those 2 sister chromatids have exactly the same genetic information on them. I think I'll leave it there for now. We'll go on to the active stages of mitosis in the next video. Other than that, thanks for checking in at MrUlrichsLandofBiology.com. Again, I am Mr. Ulrich. If you have any questions, feel free to drop me an email or put them in the comments section down below. Other than that, we'll see you in class.